Explanation of result of monohybrid cross. According to Mendel, each trait is controlled by a pair of factors. In a pure breed tall plant, two factors which control the tallness are capital T, while in the pure breed dwarf plant, two factors which control the dwarfness are small t. At the time of gamete formation, these two factors in the parent plants get segregated, as a result of which tall plant produces gametes, each containing one capital T factor. Similarly, the dwarf plant also produces gametes, each of which contains one small t factor. At the time of fertilization, male gamete of the tall plant fuses with the female gamete of the dwarf plant to form zygote, which produces progenies of the first filial generation. The process of fertilization brings two factors again together in the offspring. As a result of this, all the progenies have factors capital T, small t. Out of these two factors, the factor capital T, which is responsible for the tallness, is dominant, while the factor small t, which is responsible for the dwarfness, is recessive. The dominant gene has a tendency to mask the effect of the recessive gene. Thus, in first filial generation, although both factors capital T and small t are present, even then the expression of dominant gene is expressed. Thus, all the progenies obtained in the first filial generation are tall. When progenies of the first filial generation are self-pollinated, they produce progenies of the second filial generation. This process again involves segregation of the factors of inheritance of the tall plants having genotype capital T, small t. As a result, two types of gametes, one containing factor capital T and other containing factor small t, are produced. These gametes fuse to form progenies of the second filial generation, which have three different types of genotypes: capital T, capital T, small t, and small t, small t. The plants with the genotype capital T, small t will exhibit the tall phenotype, and the plant with small t genotype will exhibit dwarf phenotype. In the second filial generation, we obtain one plant with capital T genotype, two plants with capital T, small t genotype, and one plant with small t genotype. So, the genotype ratio in the monohybrid cross can be given as capital T is to capital T is to small t is equal to 1:2:1. In the second filial generation, we obtain 3 tall plants while 1 dwarf plant. So the phenotype ratio in the monohybrid cross can be given as tall plants is to dwarf plants.